Genetic diversity drives evolution, and evolution allows organisms to accumulate changes over time that help them survive better in their environment. Sex and its ability to shuffle chromosomes and genes to create new combinations and new organisms is a major driver of evolution in eukaryotes. But what about prokaryotes? Phenomena like binary fission are asexual. How do prokaryotes achieve genetic diversity? How do they evolve and adapt to survive their environments? In this video, we'll talk about two methods of horizontal gene transfer that do just that. Your objectives for this video are to explain two ways that prokaryotes acquire new genes and increase their genetic diversity. Before we get started, let's do a quick review from the last video. What do you remember? What surprising result did Frederick Griffith demonstrate in his work with Streptococcus pneumoniae? This was the Griffith experiment. Was it number 1, that bacterial characteristics may change when bacteria absorb naked DNA from their environment? Or was it that proteins are the hereditary molecule? Or was it that heat-killed S-type bacteria kill healthy laboratory mice when injected into those mice? It was number 1. Remember, in bacterial transformation, bacteria absorb DNA from their environment, and this DNA comes from other cells that have died in the environment. And then remember, when cells die, they start breaking down, releasing DNA fragments, which can then be absorbed by living cells. Some bacterial species only absorb DNA from others of their own species, and then some bacteria will absorb any DNA. Which of the following is essential for conjugation to occur? Is it that two bacteria must be cell wall to cell wall, or that pili mating bridges plus proteins that transfer DNA must be involved? Or is it that a super sticky glycocalyx and pili must be produced? I hope you said pili. Remember the sex pillus and mating bridges? And then the cell has to produce proteins that allow the transfer of fertility factors from themselves, the F positive cell, to the other cell, the F minus. Which of the following is a term that means to transfer DNA from one cell to another in the same generation? Is that vertical gene transfer, recombination, or horizontal gene transfer? Right, horizontal gene transfer. Great job. Now let's turn our attention to another method of horizontal gene transfer. This is called transduction, and specifically we're talking about generalized transduction. In generalized transduction, a specific virus called a bacteriophage, these are viruses that only infect bacteria. So a virus called a bacteriophage infects a bacterium. Remember back in chapter 1, I told you that one of the worst things that can happen to you if you're a bacterium is that you get infected by a virus. Do you remember why that's a bad thing? Well, when phages infect, very often they kill their host. It happens either quickly or it might take some time, but they are killing machines. In chapter 13, or when we start talking about viruses, we'll actually look at how they do this. In any case, up here, this strange structure is supposed to be a bacteriophage. They have what's called a complex capsid. And what's important to know about it is that it's a protein shell with nucleic acid inside. So the kind of magenta squiggle that you see, that's the DNA genome for the virus. So during an infection, the virus attaches itself to the plasma membrane of the bacterium. And then it acts like a hypodermic needle. It contracts and injects its genome into the cell. So you get a viral genome in the cell. That viral genome replicates via a process called the rolling circle mechanism. And I'll put a link to this in your in your lecture videos folder. So the rolling circle mechanism of DNA replication. And what's important to know about that is that you'll get multiple copies of the viral genome happening in one long piece of DNA. And then there's an enzyme that comes in and it cuts that long DNA up into fragments that correspond to the viral genome. Now, during generalized transduction, there's a mistake that happens. That enzyme that's only supposed to cut viral DNA also cuts the bacterial chromosome, generating fragments of bacterial DNA. So now we have a cell with a mix of viral DNAs and bacterial DNAs inside. Viral proteins get made, capsids assemble. Remember, capsid is the protein shell. So that assembles, and then because the viral DNA and the bacterial DNA look the same, they're not chemically tagged in any way, you'll wind up with viruses that have viral genomes, and they'll go on to start new viral infections. But you'll also get viruses that are made that have bacterial DNAs. And the important thing to know is that these fragments of the bacterial chromosome that are made, they're random fragments. So there's any number or any assortment of genes that can be on those fragments. Once this infected cell completes the viral life cycle, the virus lyses the cell, it breaks it open, releasing virus into the environment. And those viruses that are released can go on to infect new bacteria. So that's what's happening here. But this particular virus that's infecting the bacterium is injecting bacterial DNA, not the viral genome, into this recipient cell. Once this naked DNA is delivered to the cytoplasm of this cell, recombination can occur so that the viral DNA gets incorporated into the bacterial chromosome. So that's what you can see is happening here. That purple fragment from the bacterial cell gets incorporated into this new chromosome. And what this means is that this bacterium that inherits that chromosome, that inherits that DNA, it takes on new characteristics. So let's say this fragment that it inherited has antibiotic resistance genes on it. Well, then this bacterium becomes antibiotic resistant. Okay, does that make sense? So this can contribute to the evolution of these bacteria. They may become resistant to antibiotics. Could also happen that it becomes resistant to disinfectants or other strange chemicals. Maybe it picks up the ability to metabolize new substances that it couldn't metabolize before. There's all sorts of options here. The characteristics that this recipient bacterium take on is directly related to the genes that are on that piece of DNA that it got from the virus. So that's generalized transduction. Why is it called generalized? Because random fragments of bacterial DNA get packaged into the virus and then those go on to infect new cells. So it's generalized. Another type of horizontal gene transfer is called transposition. In transposition, you have little DNA cassettes called transposons that move from one location or one region of a DNA to another. And the reason they're able to do this is because you have an enzyme encoded in these DNAs called transposase. What transposase can do is it can cut the backbone of a DNA and it can ligate or glue the transposon into that DNA. So the most simple type of transposon is called an IS1 fragment, and it's simply an inverted repeat. So what's an inverted repeat? If you look at the nucleotide sequence of this strand and this one, the sequence is the same, just inverted. Same is true for the other two strands here and here. Okay, so you have two inverted repeats on the end of the transposon, and then you have this transposase gene in the middle. And this piece of DNA can basically insert itself into a chromosome and excise, cut itself out of that chromosome, kind of randomly, disrupting genes all along the way. You also have what's called a complex transposon, and this TN5 element is an example of that. Here, you have two of these IS1 elements that we see here. So we have one at each end of this complex transposon, and then in between, you might have a gene for something else. TN5 used to be used as a marker in a lot of molecular biology experiments because it confers kanamycin resistance. Kanamycin is an antibiotic that's typically used only in the laboratory to select for specific bacteria. Okay, so the complex transposon has two IS1 elements and then some other gene like an antibiotic resistance gene in the middle. So let me show you how this kind of works inside of a cell. So let's say we have a bacterium here. And as you know, bacteria have a circular chromosome in their nucleoid like this. And then very often they'll have plasmids as well. So what can happen is if a cell has a transposon in its genome, so I'm going to draw it in blue here, I hope. Nope, it didn't work. Let me try one more time. So I don't know why I can't draw in blue, so I'm going to draw it in red here. So this is my transposon here. And right now it's on the bacterial chromosome. This little cassette, because it has transposase, it can cut itself out of the bacterial chromosome and it can move to another location in the chromosome, or it could move to one of these plasmids. So let's say this plasmid now has the transposon on it. Now when this cell does conjugation with another bacterial cell, it can transfer this plasmid plus the transposon to this new cell. And guess what the transposon can do? It can stay part of the plasmid, or it could cut itself out of the plasmid and insert itself into the bacterial chromosome. Now, the problem with this is that, again, these transposons can insert themselves anywhere in the chromosome. Remember that bacterial cells are what we call haploid. So that means they have one copy of most genes. If this transposon inserts itself into an essential gene, it can actually disrupt that gene so that the protein from that gene is not produced, which can lead to death of the cell. So there can be some dire consequences because of these transposons. In a good scenario, good for the bacterium, this transposon brings an antibiotic resistance gene. And so when the plasmid comes into this new cell, it picks up antibiotic resistance that it can then pass on to other cells, increasing antibiotic resistance in a population of bacteria. Okay. But the important thing to know is that unlike the other methods of horizontal gene transfer that we've talked about, genes cannot be transferred, or let me say it again. Transposons cannot be transferred without the use of plasmids and conjugation. Okay? The transposons can move within the DNA inside of one cell, but conjugation is required for that transposon to be passed on. Just as an aside, this genetic element was discovered by a woman named Barbara McClintock. Wow, that did not come out right. Barbara McClintock. She went on to win a Nobel Prize for her work in genetics, but she was studying corn, and she was asking the question why certain kernels of corn had dots and others had stripes and still others had just one solid color. And she discovered that transposons were disrupting some of the genes that were responsible for the color in corn kernels. And it opened a completely new field of study. I think prior to her work in corn, people didn't even know that these elements existed. So what transposase does, this is just for your information, I'm not going to ask you this on an exam, but what it basically does is it cuts the transposon DNA at a specific location, which you see here, leaving what's called a sticky end. So we have all of these unpaired nucleotides here and here. The enzyme does the same thing for DNA inside of the cell, be it the bacterial chromosome or plasmid, it doesn't matter. It's going to look for specific sequence. It's going to cut that DNA. And then because you will generate sticky ends on your chromosome or plasmid DNA that are complementary to the transposon, those sticky ends will actually match up together, and then the transposase gene can simply tie the DNA backbones together, inserting the transposon into the chromosome or the plasmid DNA of the cell. And that's it. So in this video, we talked about the last two methods of horizontal gene transfer. They were generalized transduction, in which a virus called a bacteriophage, so these are bacteria-specific viruses. So this virus infects one cell, acquires DNA from that cell, and then transfers that DNA to another cell. So what you'll want to remember for your exam is that DNA is transferred from one cell to another via a virus. Okay, that's for generalized transduction. And remember, it's called generalized because random fragments of bacterial DNA are made and transferred. When we get to viruses later on, we'll talk about specialized transduction in which specific genes are transferred, but we'll save that for another chapter. We also talked about transposons. Here, DNA is transferred from one cell to another as jumping genes. These are genes that can jump from one location to another in the DNA of a cell. It's then passed on to a new cell via conjugation. So that's how cells, bacterial cells, prokaryotes, increase their genetic diversity. They acquire new genes through horizontal gene transfer. In the next video, we'll talk about another method that prokaryotes use for increasing their genetic diversity, and that's mutation. Now, you'll hear me say that the prokaryotes are increasing their genetic diversity as if this is something that they can physically do. Mutations happen kind of randomly. The prokaryotes don't have any control over that. So I don't literally mean that the prokaryotes are intentionally mutating their DNA to increase genetic diversity. I mean it more so that this is just something that happens to these cells and it increases their genetic diversity. All right, that's it. If you have any questions, you know how to contact me. I look forward to our discussions. Please continue to work on your practice exam, and I will talk with you later. Bye.